Today, I just want to give everybody a quick primer into genome topology and what that really means in terms of gene regulation and why it matters to look at. And then we'll go briefly into the techniques and methodologies behind coverage or enrichment profile on the y-axis. But we know that's not really the case. Genomes are not organized in a straight line. And over the past 20 years, a lot of really great research has been done to untangle this hierarchical structure of genomic folding. So in the human genome, there's about 3 meters of DNA in each cell, and all of that has to be compacted into a nucleus. And in order to do that, there's a series of folding events that occur. I often kind of think of this as your structural engineering of your genome and how it's all packaged into the nucleus. And so these features, these folding features, range from chromosomal territories, and then you can start getting into pockets of DNA that is more open and accessible in what are known as A compartments or euchromatin, or more compact, we're going to switch to a laser pointer, or more compact B compartments or heterochromatin, where the DNA is all kind of squished together and the histones are really kind of packed. So the DNA isn't really accessible to be transcribed. As you zoom in further into this folding, you start seeing what are called topologically associated domains, or TADs, where these are clusters of linked function genes that all kind of group together and are packaged together with inside the nucleus. And then you can start getting into individual gene loops, where you have enhancers interacting with your promoters to actually transcribe that gene. So it's very much going from that linear view of epigenetic features into a 3-dimensional view to see how things are actually interacting in the cell. And so why does that matter? So that matters because the enhancers and transcription factors that often activate the gene of interest are 10 to hundreds of kilobases away, right? So your enhancer isn't necessarily interacting with your nearest neighbor in linear space. It's acting with its nearest neighbor in 3-dimensional space. And so that's why it's really important to move beyond that 1-dimensional view of your structural organization of your genome into that 3-dimensional view, so you can start actually associating the promoters of genes for their correct enhancers or transcription factors that are mediating gene expression. So how that folding all occurs really does influence the gene expression that you see within a cell. And so this matters in terms of humans and diseases and gene regulation by looking at whether or not these 3-dimensional structures or these folding patterns are disrupted in different cell types. So in this really great study that was published in 2019, they were looking at why an oncogene, a cancer gene, was being activated in this particular study, and they found that a over-methylation site actually destroyed this gene loop here. And so in the normal scenario, this super enhancer that normally does not interact with this oncogene, right, is being separated. So this acts as a barrier between gene activation. As this loop gets broken, this enhancer is now enabled to activate this oncogene and therefore driving dysregulation and leading to a cancer phenotype. And so understanding how your enhancers are interacting with your gene promoters is very, very critical in understanding how genes are being activated, right? It's not just that they are activated. In order to really understand that phenotype, you have to understand the mechanism of action. And that 3-dimensional folding is a means by which to describe that mechanism that turns the gene on or off. And so how you would look at 3-dimensional genomes is a very unique approach called proximity ligation, and there are various flavors of that. we have sort of this all-in-one solution which provides really uniform coverage that enables researchers to interrogate your single nucleotide polymorphisms, because we get really great coverage, and we'll show you why in the next slide. You can interrogate large structural variation, and you can use that SNP information to get allele-specific features, so you can do your phasing approaches as well as capture the chromatin topology. Now, if you're only interested in a single region here, this is actually very compatible with hybrid capture. So if you're interested in, say, oncogenes, you can get an oncogene hybrid capture panel and throw it at one of these libraries and ask what are the interactions that are associated with your gene of interest. And then the two that we're really going to dive into today are this micro-C approach, which really gets you down to interrogating chromatin folding at the nucleosome level. And we'll show you why that is here in the next slide as well. And then when we combine that with chromatin immunoprecipitation, you get what's called a high ChIP, which gives you your ChIP signal, your enrichment over DNA protein binding sites, and the long-range interaction. So the sort of base of the data that we're going to be showing today is this micro-C or MNase digestion. Won't get too much into the details here, but really what this involves is if we have nucleosomes that are linked together by linker DNA, what we're doing is we're cutting the DNA between the nucleosome and chewing back, so we get these nice, very uniform fragment sizes. And then once we have them cut, we have everything ligate back together in three-dimensional space. And so you can start seeing which nucleosomes interact with each other. And so what that gives us is this nice coverage profile where you can interrogate, nucleosome position, as well as what nucleosomes are actually interacting with each other through either short-range or long-range interactions. Now, historically, this approach to interrogating that 3D structure, the chromatin was fragmented at restriction sites, which generates much more random fragment sizes and fewer possible ligation events. So you're capturing fewer of those folding events, and your coverage per each one of these fragments is actually being split because you're enriching your reads right over a restriction site rather over a chromatin-centric feature like a nucleosome. And so if we take this approach and want to interrogate a particular protein DNA binding site, such as H3K27 acetylation for enhancer regions, or H3K4 trimethylation, which is indicative of active promoter regions, we can do this proximity ligation and then pull down on these particular histone modifications to give us a very unique view of the genome, as giving you the primary binding site and the interactions that are associated with that particular histone modification or protein DNA binding site. And so the workflow for these sort of assays, which is very much the cornerstone to the talk that David's going to be giving here very shortly, is that we start by fixing cells in three-dimensional space, right? And once the DNA is fixed, then we go ahead and we fragment. We use, I just mentioned MNase to do that fragmentation to give us those nice uniform fragment sizes. And then we conduct our chromatin immunoprecipitation. So we pull down with any antibody of interest. And then once we have the protein and DNA complex isolated through the chromatin immunoprecipitation, we perform ligation of free ends, giving us that long-range information along with the protein DNA binding site enrichment. We reverse those crosslinks, giving us chimeric DNA that's reflective of that 3D orientation. And then we generate a sequencing library which results in the ability to interrogate the 3D folding and to kind of move away from that linear space model. So here's what the data look like. So here what we're showing is normal ChIP-seq data. So again, just looking at where the DNA protein binding sites are for this particular CTCF protein. And as you can see, there's enrichment along the genome for these DNA protein binding sites. But what you don't get is what is interacting with what here. And so with the high chip approach, bringing in that 3D information, we can recapitulate that chip signal, that protein anchor signal, but now we can see that there are sites here that are interacting with each other a lot more frequently than, say, like this site interacting with this site over here, but this binding site interacts greatly with this region here and this region here, indicating that these are chromatin loops isolating genes within this region. And so if you zoom even further onto these peaks, because we use that emanase to fragment, you can now position your nucleosomes around that DNA protein binding site and not just look at where the DNA is enriched. You can actually look at where the nucleosomes are positioned around that site. So again, those are just sort of the three different flavors of how you would go about interrogating the 3D genomic structure, that genome topology. And so with that, I would like to say thank you and get over to our main event. So I'll pass it off to David.
Thanks, Corey. I'll just share my screen here. Hi, everybody. All right. Are we seeing that okay, Corey?
Yes.
We're good? Okay. Hey, well, good morning, everybody. It's great to be here. Most important part on this slide is that I am a consultant and I work with Dovetail Genomics as listed down below. That's my disclosure. It's been wonderful to see the company grow from over the last 7 years to where it is today. So what I want to do today is to talk about how we've collaborated with Dovetail Genomics to have a much better mechanistic understanding of, if you want to boil it down, why do we age? And then I want to end with some work that is more about perhaps why we don't have to age as quickly as we do. But first of all, I want to talk just a bit about aging. What is it? Well, everybody thinks they know what aging is. This is a good example in this slide. This is me looking about 30 years ahead. And we all think we know what aging is because when we see it, we know it. But we know very, very little about what drives this process. We know that there are about 9 main drivers of aging: telomere shortening, mitochondrial dysfunction, protein aggregation, and the list goes on. But one of the so-called hallmarks of aging that has and is rising in interest is epigenetic alteration. And the more my lab and others around the world study these changes, the more we're coming to an appreciation that these changes may be primary or major controllers of these other events that drive aging, such as mitochondrial dysfunction and protein misfolding. And so I want to tell you about the work we have done in the last few years and about a paper that's now in press coming out. Anyone watching this probably knows a lot about chromatin, but if not, the types of modifications that I'm going to refer to are up in the top right: H3K27 acetylation, which will open up chromatin, as well as H3K27 trimethylation. These are marks that will open up chromatin, desilence genes, and make them poised to be activated. And this is extremely important for development. Otherwise, we'd just be a mass of stem cells, world's biggest tumor. But because every cell can define its own cell type and function in a specific way, thanks to, in large part, this process, we are born, we're healthy, majority of us are healthy, and we stay healthy until you reach about my age. I'm 51. And then things start to go wrong. And what we're learning is that one of the main things that goes wrong is this system that is set up early in life, but eventually is disrupted. And so the big questions that we ask are, well, one is, why does this occur? Second is, if it does occur, does it drive aspects of aging and disease in mammals. We know it does in yeast cells. This is what I used to work at when I was at MIT with Lenny Guarente. We know epigenetic change is a major problem during aging of yeast. Hence, one of the reasons we work on sirtuin genes is because they were first found to control longevity in yeast cells, and silent information regulators, or sirtuins. The word information and silencing is in the name. But now what we've come to realize is that the silent information regulation, including the sirtuins, are important for these changes that happen during aging and may actually drive aging itself. And then the question is, if this happens, is there information in the system to reset so that you could literally reverse the aging process? Waddington's landscape, 1950s, Conrad Waddington had this metaphor for how a cell develops into its specialized forms. We had Corey tell us about how this happens at the molecular level. We have tools that Dovetail provides as, that allow us to actually see these loops and structures and bundles, one of which is called a TAD. And what I hope that I drive across today is the realization, increasing realization, that aging may be due to the changes in three-dimensional structure of the nucleus of chromatin, and that cells actually drift along the Waddington landscape and start to lose their original identity and therefore their function. So think of it as a nerve cell in a young 20-year-old is functioning perfectly. They can remember most things, but by the time that person becomes 80, those nerve cells in the brain or at the back of the eye have partially forgotten that they're actually nerve cells and start to take on chromatin that resembles other cell types. We call this process epigenomic noise, and it's similar to scratching up a DVD so that you cannot read the information, which in biology would be the genome. I'll show you a few slides on what we think is a major driver of these epigenetic changes over time. Before I do that, I just want to have a shout out to Claude Shannon, who was at MIT in the 1940s. And this is from his paper in 1948 about how to have lossless transmission of information, which gave rise to the internet. The internet is basically built on this wiring diagram and math in this paper. But I think it's relevant to aging because there's a source of information, there's a fertilized cell. A transmitter would be our body when it's young. We have a lot of noise, which I'll tell you about. And then the receiver, in this case, would be our bodies in the future. And the information that was once in our young cells has partially been lost over time. But the question is, do we have a backup copy, which Claude Shannon called the observer? Today we'd call this the backup, or the TCP/IP protocol that makes sure that emails and photos make it across the internet. Claude Shannon said, well, if you have a backup copy, you can correct this information and reassemble the original message that was transmitted. And that works great for electronics, but can we do this with biology? Can we take an old tissue and reset the switch and correct the epigenetic data? And what we're finding is that it is possible. This is just a different way of representing this, that genes that are on when we're young seems to be switched off, and vice versa. In this case, the sirtuins that we work on, the silent information regulators, are part of this process where a silent gene, let's say down here, where would it be? Well, here's a good one. Here's a silent gene. Sirtuins are controlling this silent region. And then over time, we actually saw that those sirtuins get distracted in large part by double-stranded DNA breaks, and then they move away. And in an old cell, genes will now be switched on that really shouldn't be switched on. And that gives rise in part to the aging phenotype, such as sterility of those old cells. So we have been looking at this in mammals to see if broken chromosomes, these DNA double-stranded breaks, are a driver of epigenomic noise and aging. So we built a system, full credit to Philip Oberdorfer, who's now at the NCI. We started making this mouse a decade ago. So we made this mouse where we could induce an endonuclease from a slime mold called PP01. We used it because it was previously used to study DNA repair, and it doesn't cut very often in the mammalian genome. Per cell, in practice, it only cuts a few times. There are more recognition sites, of course, but due to chromatin, it doesn't cut at every recognition site. So now we could induce slightly above background levels of DNA breakage. Of course, DNA breaks are happening all the time in our life, at least one per cell per day, which is a total of 26 trillion in our bodies. And a broken chromosome is a disaster for the cell if it's not repaired. And there are large-scale changes within the cell of moving proteins, such as the sirtuins, to the break, and then they have to find their way back to the silent regions again. So it's this dual use of chromatin factors and double-strand break repair factors. The same proteins are involved. And that's our hypothesis, that if you do this enough, this movement, that eventually some of those proteins that go to repair don't go back to where they came from, and you eventually disrupt the epigenome. So we have these mice, we call them inducible changes to the epigenome mice, or ICE mice. They have nothing to do with border control. And then we can also pull out cells from the mice, either embryonic or adult fibroblasts, and do the same experiments. To cut to the chase, I want to tell you what happens when we create a few of these non-mutagenic breaks. We don't leave mutations, that's important. And we also don't disrupt genes that are around the break. It's actually changes to the genome, epigenome happen very far apart, megabases away, primarily because of this relocalization. So this mouse was treated with the system, but it lacked the enzyme, so it didn't experience any additional DNA breaks. So its epigenome 10 months later looks just like a regular middle-aged mouse. And you can see it looks pretty healthy. We treated for 3 weeks, raised the double-strand break level, and let them go back. At that time, there was no difference. They didn't feel it. They didn't get sick. It's a bit like getting an X-ray. But the mouse that received the DNA cuts 10 months later looked like this. And we've done hundreds of these experiments now over the last 7 years. And these mice don't just look old at the histological level, physiological level. At the molecular level, they look old. And I'll tell you a little bit about how we've done that, which we're in the process of publishing a couple of papers on this. Jae-Hoon Yang is the main postdoc who's working on this. We can also now look at the age of these mice with less subjectivity by looking at the DNA methylation clock. We can also model this mouse in vitro, this system. These are, in this case, MEFs, embryonic fibroblasts. And what we do is we induce those DNA breaks for 24 hours, slightly above background, about 3 to 4-fold. We let the cells recover, and they grow normally. And we can look at things like, do they have mutations? The answer is no. Changes in cell cycle? No, not during any of this process. And what about the epigenetic information? Does RNA-seq change? And do the contacts between areas within the nucleus change? And this is where Dovetail's technology has come in. Interestingly, if you go to a textbook or even literature right now, people would say, scientists would say that this cell is no different than this cell because it's repaired the DNA. But very rarely, if ever, do scientists consider that having gone through the process of DNA repair, there might be changes to the epigenome. And I can tell you that there are dramatic changes to the epigenome, even if there's no mutations. So this is a high-C topology map across chromosome 6. And just blowing up one of these regions, you can see that in 3 repetitions, biological replicates, we see that there are changes in chromatin contacts in some regions. Just to repeat, this is not where the DNA cutting was. This is not a PPO1 cut site. It's some other sensitive region for reasons we don't fully appreciate just yet. The other thing we can do is RNA-seq, and I won't show you the whole data, but those RNA gene expression changes look a lot like aging in the animal. And the kind of processes, hopefully you can read on your screen, look a lot like gene sets that are turned on and off during embryonic development, which is interesting because our thesis is that cells will start to de-differentiate during aging. Or actually, we refer to this as X-differentiation. It's a smoothening of the epigenetic landscape rather than having very defined regions. And one of the regions that we found that was highly dysregulated overall for a variety of modifications to histones was this cluster, the Hox locus, which you'll remember controls during development your head from your tail. You do not want to mess this up. And you also do not want to be promoting cancer in an adult. So this is not a region that you want to dysregulate. Nevertheless, in aging and in this eye system, we do see that these genes are deregulated. And this is a standard analysis. This is ChIP-seq looking at changes to H3K27 acetylation. The eye cells are in red, so you can see here the acetylation levels are decreasing. Same for K56. And a lot is going over here on the distal end of the Hox locus. You can see these are all the Hox genes down here in black. Those are the reading frames. And so we're losing a lot of H3K27 trimethyl as well. So we're starting to desilence at least this side of the locus. You can see an RNA-seq down here. These genes over here are coming on. And interestingly, over here, this proximal part of the Hox locus is being silenced. So here's where a high ChIP came in to help us understand what's really going on. We got so much more information than the regular ChIP-seq. Now this is serving as a negative result. We looked at those cut sites where the enzyme was cutting, and Dovetail and we found really no changes based on high C or on high ChIP. In this case for CTCF, the TAD boundary protein, and K27 acetyl and trimethyl. Okay, and again, well, I'm not showing this, but in terms of RNA-seq, there's really no significant changes. But when we look at the Hox locus, things really emerged as being altered. So this is, first of all, one cluster of genes called the HOX-A, and next I'll show you the HOX-B. Here, the Dovetail team has highlighted in red changes or contacts between promoters of Hox genes to non-promoters. So that there's the gray, which are the contacts, and then the red is just to highlight those other contacts. And what you can see is that the control cells that were not cut, this is just the Cree control, these are basically just passage cells, they have a regular type of contact between various regions within the proximal Hox locus. But if you cut the cells with this PPO enzyme and let them recover, you might say, okay, those cells look pretty similar. But at the chromatin level, there's huge changes. You can see that these genes here, HOX-A13, for example, is now making contacts with these new sites, perhaps enhancers up here, that are leading to changes in transcription immediately or perhaps further down the line as cells become more and more senescent or closer to senescence. And you can see that these changes here in the eye cells have a big effect on the actual chromatin modifications and on the gene expression down here in RNA-seq. And this is information that you wouldn't get if you only did high C. Now for the HOX-B locus, it's a little different in that there are a lot more contacts in the untreated cells. And here you can see this is a cluster that's making contacts with these new enhancers that we identified in this study. But these are lost after the in vitro aging of these cells, which is really quite a remarkable detail to be able to be picking up with these experiments. And so we were really delighted that we could finally see at such detail of how the chromatin is changing in its organization. And that helped explain the changes both in the bulk modifications, but also the gene expression patterns that we think are leading to a loss of cell identity and aging. And this is just a model that we have in our manuscript. And again, so double-stranded breaks cause proteins to leave loci, such as the sirtuins. We think PRC complexes are important. And then if they don't fully find their way back to where they came from over time, you get this deregulated chromatin, which is not healthy for the organism, and we think may drive aspects of aging. If you want to look up some of this data, we've put it up on bioRxiv. DNA methylation clocks, you may all be familiar that you can train with machine learning methylation patterns that tell you or us if a mouse or a human is older for their age or younger. When we looked at those mice that I showed you, many of them were older, on average 50% older than the control mice of the same litter or the same age. And the actual removal of DNA methylation is controlled in large part by these TET enzymes. These are, think of them as DNA demethylases. And there's another one called TDG. And so I'm going to show you later a bit of data about TET enzymes, whether or not TETs are involved in resetting of the clock, which we think they might be. But the first question is, can we take cells or mice that are old and make them young again based on the clock? And if we do that, does anything happen? Do the mice actually get younger? Do they get healthier? Or is the clock just an association or an epiphenomenon? And this is a big question in the field. And I want to give a shout out to the folks that have helped us with the clock. Includes Vadim Gladyshev, Margarita Mir, Steve Horvath. We've had a lot of help from Morgan Levine lately, and Zymo. So the last part of my talk is about reprogramming tissues to be younger again. We were looking for ways to reset chromatin. In other words, try to find the observer that Claude Shannon's proposed might exist in his mathematical equations. And we looked at lots of different genes. Wencheng Liu, the former graduate student in the lab, spent about 3 years trying various oncogenes, transcription factors, trying to get a reset of the clock. And he was inspired by Juan Carlos Belmonte's work. It was a Cell paper in 2016 where they had turned on 4 of the Yamanaka factors, which you'll know are usually used to generate iPSCs. But what he did with his postdoc, Ocampo, Dr. Ocampo, they put all 4 genes into a transgenic and turned it on in a mouse. So the bad news is that those mice died after 2 days. c-Myc is one of the genes in there. So really not too surprising. But what they did was clever. They stopped the genes after 2 days, turned them off and let the mouse recover for another 5 days, and then repeated that. And a short-lived mouse lived 40% longer and seemed to be healthier. And that was the first evidence that you could turn on reprogramming factors in a mouse, albeit very shortly, for short periods of time, but perhaps make them healthier. But there was a lot of skepticism. Maybe just stressing a mouse and almost killing it will actually make it stronger in a form of hormesis. But we thought, this looks really interesting. We had some success with Yamanaka, but we always got teratomas because c-Myc is in there. So what Wencheng decided to do was to leave out c-Myc and just put in the 3 other Yamanaka genes, OCT4, SOX2, and KLF4, OSK. He managed to cut down the size of the adenovirus, adeno-associated virus, AAV, and fit them all in one cistron so they could all be expressed within one virus. And then he infected cells in the dish, fibroblasts, and could see that we could reverse the age of those fibroblasts. But then he did this experiment, which was a really brave and clever one. When you're very young, let's say you're a neonate or an embryo, if you crush the optic nerve or damage your spinal cord, it's not going to grow back. But it could grow back, but if you're our age, it's not going to grow back. And so he thought if you make the optic nerve very young, by reprogramming it, it might actually grow back, and that could be a great assay. So we teamed up with Zhigang He at Children's Hospital in Boston. This is where the crush is. On the right-hand side is the eye. On the left-hand side is the brain. And this is if you don't if you don't treat those nerves, the dye will not progress down here because the axons have died back. And a lot of the nerves in the optic nerve have died. These are RGCs. But if we turn on the OSK factors a couple of days after the crush, this was our first result. You preserve the neurons, and some of them we see start to grow back towards the brain, and the longer we leave them, the better they grow. Now, we also treated glaucoma in mice, and we were able to partially restore vision in those glaucoma mice. But this is I think the most exciting experiment. If you just take regular old mice that have lost their eyesight, Just ignore the feces. That's just because the mouse is a bit nervous. But what we're trying to see is the mouse should move its head down the screen in response to those lines. And this mouse is functionally blind. And we can also measure the electrical signals at the back of its eye, which we have, and those signals are extremely weak, if not absent. But if we reprogram the eye by putting in AAV and turning on reprogramming factors, we get old mice that now can see again, just like they were young. And this is an example of that mouse. And when we look at the electrical signals in the optic nerve, they are behaving just like a young mouse would. So we've gone from improving the survival after injury, and now I'm showing you the improvement in function due to regular aging. And interestingly, we find that those processes are quite similar at the epigenetic level. In other words, the DNA methylation changes that occur after injury are highly significant in their overlap with what happens during aging, and that perhaps the OSK reprogramming works because it's reversing aging in both those situations. Well, is it reversing aging? We can test that. One is to look at the transcriptome, and if nothing happened to those cells after reprogramming, basically you'd have a flat line. If it accelerated aging, those dots would go from the lower left to the top right. But if it reversed aging, you'd get from the lower right to the top left. And I think you can see that very clearly the gene expression changes do look a lot like they are going back to being a younger state. And the blue dots are the significant ones. The red ones are also highly significant, but I've colored them red because they're interesting. They're involved in sensory perception. And why sensory perception and taste receptors in particular are changing in the eye, we don't know yet. But we just wanted to point that out, that this group is particularly interesting, and we're going to follow up why these would change during aging. The other thing to note that may not be obvious is that if you look here, that genes that went down in aging, okay, here, down in aging, so they're going to the left. If they go down a little bit with aging, they go up a little bit with reprogramming. Those that went down a lot with aging go up a lot with reprogramming. So in other words, the reset doesn't only know which site or gene to fix to be young, it also knows the amount of expression that it once had. And these changes depend upon the TET genes. So here they are. These are the enzymes. There's TET1, TET2, TET3 that demethylate DNA. And I mentioned TDG, which is important for active demethylation, not just passive due to replication. And we find that TET1, TET2, and TDG are necessary for this epigenetic reset and the recovery of function in the eye. And this is an example of that. If we look at the movement of the mouse's head, called optomotor acuity, we can improve that with the treatment, and it doesn't work if we knock down the TETs in the eye, at least TET1 and TET2. So that's pretty interesting. That means that perhaps the TETs are part of this reset process. In the case of the injured eye, we can also look at the age of the cells. We see that injury increases the age, and then we can bring that back down again with our OSK treatment. And if we look across the entire methylome, not just at a few sites, this is on the left would be, I think it's based on close to 100 sites. If we look across the entire methylome, and now we color this in a heat map, you can see that injury changes the DNA methylation patterns, and that the OSK reprogramming largely brings that back. And this was done with Morgan Levine and Margarita Mir at Yale University. But the question is, how the heck does OSK, transcription factors, know which of these genes to reset and at what level? How does it know that these genes, or this site, let's call it methylation site, needs to go back to this level? And similarly here, there must be information encoded in the epigenome that allows for the cell to find this information and reset. So this is the model that we have changes in methylation due to both aging and injury that are similar. We have evidence that the sites that are changing are overlapping with the PRC1 and 2 complex, which is involved in both histone and DNA methylation changes. And that if we don't have TET1 and TET2 or TDG, this process of reprogramming to be young again does not occur, demonstrating that they are necessary. And perhaps experiments show so far that they're not sufficient. So they're part of the system, they're part of the clockwork of resetting the age of complex tissues to make them functional again. So this is the model. These are some of the changes that we see downstream and we're looking at. First, I've told you is double-stranded breaks can accelerate aging, including the DNA methylation clock. Epigenetic reprogramming, in this case by O, S, and K, can reverse the clock and restore youthful function of a complex tissue, such as the eye. Reprogramming requires active DNA methylation. And perhaps this is a supposition, but this points to the possibility that DNA methylation changes may not just be a measure of age, but actually a participant in the aging process and a requirement for the reversal. And then finally, if we go back to Claude Shannon, I've told you that DNA damage, double-strand breaks, is a source of noise that leads to the loss of information in old cells, that OSK is part of the reprogramming and the reset. We know it's part of the correcting device, the TETs and TDGs, perhaps PRC2 complex. But the big question is, where's the information stored for this reset? Is it in the DNA? Is it a protein that binds to modified histones? Is it something else? Is it a DNA-RNA hybrid, or even some DNA modification that we really have overlooked for the last 50 years? So that's what we're working on. I have a lot of people to thank. In my lab, the people highlighted did a lot of the work that I told you about today, and I aim to highlight them as I was ongoing. And I also have a lot of collaborators that I want to thank. And in particular, for this talk, I want to thank the group at Dovetail Genomics that did an incredible and speedy job getting that data for us that we otherwise would not have been able to do, and give us a real detailed look at what might be going on during aging and then how to reset the system. So thanks, I'll end there and take any questions and keep this slide up a bit to thank the people that funded the work as well. Thank you.
Great, thank you so much for your talk, David. So we do have a number of questions. We'll start just going down the list. Here we saw histone modification, MET and ACID. What about influence of a phosphorylation?
Great. We have not looked at phosphorylation at all. That's totally up for grabs. If anyone would like to look at that, I think it'd be a great thing.
Awesome. CTCF binding is affected by CB methylation. So is chromatin contact changes in HOX loci due to the CB methylation?
We don't know that either. We didn't see dramatic changes in CTCF by ChIP or even by high ChIP. So we suspect that the protein itself is not moving from sites, but perhaps is changing in its modification and association with other proteins. And that's something we'd like to test.
Great, thank you. How long, so this is a question about just how long these changes last. After reprogramming, do you require a permanent stimulus or treatment, or is it enough with a certain amount of time?
We've looked at the eye reprogramming, and the preliminary data says that it's a permanent alteration, which you would expect if DNA methylation is changing. DNA methylation is very stable. But yeah, we haven't done extensive studies of how permanent it is. If I was to bet, I would say it is a full reset, like an iPSC would be a reset. And then you just age out again. But we really have to test that. It's just speculation. The other question that I think is interesting is how many times can you reset a tissue? Is it once, which we've done, or is it 100 times? We don't know that.
Okay. All right. Do you think this could repair early onset natural glaucoma in mice?
I think vision loss, if it's affecting the RGCs, the retinal ganglion cells, yes, I think it could. That's what everything points to. Our goal is to use this AAV method to treat glaucoma in general, whether it's early or late. And I would expect results in about 2 to 2.5 years in the first clinical trials.
Mm-hmm.
So yeah, I think it's quite feasible if it's due to RGC dysfunction.
Okay. This question is regarding the reprogramming of the cells, the optic nerve. How long were those results seen? If you answer that, then there's a bunch of other little questions to this question.
Oh, how long did we see that
The
effect?
results with the optic nerve.
Well, I think I understand the question. That first experiment was after 3 weeks, but if we go for 16 weeks, those nerves will actually reach the brain and start to make connections. Not all of them, but a few. We haven't tested vision in that system. We just think it's unlikely that the mice would get their vision back. But if we learn to do it even better, say, use rapamycin in combination with our treatment, it's feasible we could make those mice see again. And that's something that we're interested in doing. But yeah, it's somewhere between 3 and 16 weeks seems to be optimal for that experiment.
Okay.
Oh, and also we've done it in old mice, not just young. So the field of nerve crush uses really young mice. I think it's 5 weeks old or something. No, even less, 3 weeks. That is because that's where you get the best results. But we took 1-year-old mice and it still worked. Not as effectively, but it still worked.
Great. So looks like people are to the audience who are submitting questions. Please make sure you are submitting them in the Q&A module as opposed to the chat. Then it's easier to keep track of the questions. So next question regarding the clockwork or reset, or can you reset the clockwork? I'm just gonna. Is the clockwork of resetting seen in cancer?
Gosh, that's a great question, and shame on us for not having done more of that to see what the overlap is. Now, there should be some overlap because the cells are getting younger and cancer cells are generally young. But here's the good news is that we've overexpressed OSK for over a year in mice, systemically, not just in the eye. We've injected the AAV9 system and turned it on, and those mice are perfectly healthy. We sacked them because we wanted to look at tumor burden, and the trend was towards lower numbers of tumors, not more. And I suspect the question is also related to that concern. What's interesting is that if you use OSKM, you will get stem cells in the dish. This is what everybody does in the lab. But leaving off c-Myc, the M, we've been very lucky. I think this is the reason why it's so safe, and that there's this apparent barrier so that cells go back in age, but they stop and they don't go towards being cancerous. And we know this because we've also looked in the mouse and in cell lines for the expression of cancer indicators, such as NANOG, or indicators of transformation would be the right word. And we don't see it. NANOG is, if we start to see NANOG, we know that we've overdone it, and we did not see that in our experiments. And that was one of the early things that we did, because the last thing we want to do in treating animals, and especially humans, is to start to promote oncogenic transformation in our in our therapies. So we're very lucky. And here's what I think is going on. We've tapped into a very ancient system that salamanders and lizards use to regenerate tissues and can survive. We also can regrow new liver if we cut half of it off. But for most of our tissues, such as the eye, we've evolved away from the ability to regenerate, and that here we're restoring that ability.
Wow. in your genetic switch on and off reprogramming system, have you looked at or have you been concerned about the influence and effects of the immune system changes?
Not enough. I would love to know if we can restore the immune system using this technology as well, because I appreciate how important that is, not just in the pandemic, but for aging as well. Mice that have defects in their immune system, T cells in particular, will age rapidly. So yeah, it's a good question. We just haven't looked yet.
Okay, thanks. I understand the process of injury, but how does aging affect methylation, which is clearly a slow and gradual process, maybe even stochastic?
Well, I wouldn't say it's stochastic. Some may be, but the fact that we can build clocks and see reproducible changes and reverse it means that some of it is not stochastic. There are HUTs for DNA methylation. It's also worth clarifying that we see demethylation changes as well. That's a bit of a poorly understood point. So I think the question is, how does methylation change with aging if it's a slow process? Well, what our model is the following, is that damage to the cell, such as double-strand breaks of DNA, but there are presumably others that stress the cell, require the unpacking of chromatin and the movement of chromatin factors to other regions in the genome. And that's part of an advantageous stress response that turns on repair genes and other changes, say to metabolism. But the resetting of that information, getting those proteins to go back, to get the DNA methylation changes, if they occur, to go back to where they were after the repair and the emergency is taken care of, is not 100%. And so that slowly, day by day, the cells are accumulating small changes to where transcription factors are, how histones are modified, and how DNA is methylated. And by the time you get to my age, you can start to see the effects of that. And then it starts to get exponential, because it's a positive feedback that once you start to have a dysfunctional epigenome, you have less and less ability to reset the system accurately.
Yeah. Can these changes transfer to offspring?
We don't know that. Now, there's a lot known about epigenetic transfer of the environmental changes. A fat rat mom will give rise to pups that are predisposed to diabetes. But we haven't done that great experiment, which is to accelerate aging in the egg or a sperm and see if that mouse will have signs of aging earlier. It's a really great experiment.
Yeah. Have you looked at other heterochromatin markers? This person asked about specifically H3KM or H3K9ME3, but maybe other ones?
I'm not sure if we've looked at that modification. I don't recall that we have. And so now what we're doing is going through the list of key changes and repeating these experiments. I think that's the next really good idea, which is to build up a three-dimensional, or actually in this case, four-dimensional map of the changes that occur both in the cell-based system and the animal, so that we can understand in a time series which steps occur early, which steps occur late, and then reprogram the cells to see how it all gets unwound.
Mm-hmm. There is some evidence that DNMTs may be recruited by DNA damage repair complexes. Do you think this may contribute to—
Right. Yes.
Yeah,
it's a good idea. Yeah, when we look at our genes that are involved in aging, and we think reprogramming, you can find connections to DNA repair as well, which would fit beautifully into our model, which is that the cell has evolved to use the same proteins that are involved in development and aging, also for DNA repair. And that's our Achilles heel. And it's adaptive because when we're young, it's good to coordinate gene expression with DNA repair. But over time, it really doesn't do good things to the epigenome. And I think DNMTs are very likely to be involved in the same way that the TETs are the yin of the yang. And so, yeah, we should do that, is to knock out various DNMTs and try to reprogram and see if that works, among other experiments that I think would be useful.